Okay. Good morning, and thanks for coming to the UMGC's presentation at your seminar series. I have a provocative title because I wanted to get people thinking about the future of sequencing, a future that might be, 2 to 5 years off, but the one thing I should say is that this is, of course, an absurd title. Sequencing will never be free. Just wanted to get that out there. And we've been spending a lot of time talking to the company Ultima recently because we've been using their new sequencer. It's the most recent acquisition of technology. And I was mentioning to them that yesterday I was talking to somebody who wants to do a lot of whole human genomes. Human genome sequencing 30x coverage, used to cost thousands of dollars. And I mentioned that it was going to be $226 all in. And she said, well, but I thought it would be less than 200. And I thought, oh my God, even 226 is now not considered cheap enough. And that Ultima said, just remind people it's never going to be free, but it could be close to free. And that's the overall topic I want to discuss today. So I want to start with a metaphor, I think almost entirely in metaphors. And so this metaphor I'll use is computers. Why do we call these, for those of you online, I'm holding up my phone, this is a computer. We call them computers because once upon a time, they were people who did computation. And you can imagine, since doing computations with people is really slow, computing was really expensive. These were people who sat there and just did math all day long. That slowly gave way to machines doing it. And in the early days, the word computer made sense because they still were doing calculations for explicitly calculation-driven endeavors like engineering, military applications, space travel, So they were recognizable what the function of the computers was, and the application were still pretty much one and the same. That started to change at the time that these things appeared on our desks, because now all of a sudden you are using it to write, usually, and some other things. And of course, this is now your favorite computer. And what it does is so removed from computation that the word computer is really, it's a holdover. But in actual fact, that is what those things do. Just tons of computation all the time. And it's invisible to us now. And that's the point I want to make about sequencing. So what do you do when computation becomes basically free, which it is more or less for our phones? I mean, the phone's not free, but the computation part's not the really expensive part. You can do lots of operations. And the things you do are from the sublime to the ridiculous. Sublime, this company, Pixar, that does these incredible movies, came out of Steve Jobs, when he left Apple, had this company that had a thing called RenderMan, and it was really meant for engineering design. And then they realized, well, actually, it's great for making images. And they realized computation was getting so fast, you could actually just turn that company into Pixar, which is what happened. The lower picture there, cars, I don't know if you're not maybe geeks in the same way, but that movie used a thing called ray tracing, where a computational engine simulated every light in, for instance, this NASCAR stadium, and had the light bounce around off every surface that had been modeled. And that's how these kind of like images you see on the front of those cars, that's where that came from. It's not like somebody's painting in reflections. They calculated and simulated the world. And of course, you also get this kind of stuff. It's another outcome of computation, unfortunately, but that's the same thing. Behind everything in the modern world, computation is going on all the time. And when it's invisible, a couple of principles I think are relevant. One, as the input into a system, in this case, whatever system a computer is built into, becomes arbitrarily inexpensive and pervasive, it tends to be used in a wasteful fashion. Like, you don't care about the cost, so that just becomes useful. And I just did this last night. I asked ChatGPT to give me an illustration that conveys the wastefulness of energy consumption by your AI computation, and it cheerfully made that little rendering. That apparently uses like the amount of energy to heat up a cup of coffee. So I apologize to my future grandchildren and great-grandchildren for this, but it is entertaining. But that is just tons of math that went on in the background, right? but I don't think of it as math. I think of it as, AI, whatever. The other thing is that as the input into a system becomes arbitrarily inexpensive and pervasive, other inputs become limiting. That's actually highly relevant to us today. And an example of this is that all of these tech companies are now starting to fund nuclear power plants because although the computation is cheap, the electricity is actually the limiting factor. Like, it's not hard to make silicon wafers, but now the electricity, which wasn't the problem in the past, is the expensive part. And so they're pivoting to, like, my God, where's the water? Where are these inputs coming from? Okay, so the relevance to us and you is, as pure sequencing becomes arbitrarily inexpensive and pervasive, we'll use it in a wasteful fashion, and other inputs into our scientific enterprise will become limiting. We're focused on those two things. One, how do we help you start to use sequencing wastefully? And what does that mean? I mean wasteful in quotes, because obviously you don't want to waste your time, but oftentimes it makes sense to waste sequencing. And so this kind of concept of sloppy seq is what I'd like to coin this. Sloppy seq is that you stop spending a huge amount of time worrying so much about what sequencing costs and allows you to do other things. So to make sure that I don't forget to give answers, I'm going to start with answers and then go into the technology that enables them. So these are some thoughts that we're having in the Genome Center right now. Enough with sequence capture. The idea is that for people who are looking at genomes, sequence capture is now past its prime. The whole genome is the unit of analysis. So, for example, recently we had a client come to us and they wanted to sequence a 4 megabase region in 1,000 samples. And we knew that the Ultima was coming, the UG 100, and we said, we can actually give it to you for less money to sequence the whole genome, because the sequence capture probes are going to cost more. And all of the complications of that kind of project, like they wanted to add little content. So there's like a week or 2 months of waste while you talk to Twist about the content. All of that complexity you can just get rid of by saying, let's just sequence the whole thing. Another example where Mike Gale is coming to the U. I don't know if people know of him. He's going to be the head of Department of Microbiology. He has very large projects where they use whole blood RNA and they do whole blood RNA-seq. The problem, of course, being that most of your reads, when you do that on whole blood, are globin from the erythrocytes. And so everybody does globin reduction to a tune of $75 to $80 per sample per capture to just pull out globin so you can go ahead with RNA-seq. And we proposed to him, and are doing an experiment right now, what if you just sequence all the globin and just throw the reads away? Maybe you can even throw them away at the time of mapping. You'll save $80 per sample. You're not going to have whatever errors are introduced by that globin reduction hybridization working differently in each sample, and you get the same data you want, and it's faster. So that's what we're trying there. Another example would be PCR. A lot of the time you do amplification or capture to get viral PCR, but if the sequencing arbitrarily cheap, just sequence everything that's there. If you're interested in somebody who might have a viral infection and you don't know what it is, take the cerebrospinal fluid and sequence everything in it, all RNA and all DNA. Bulk wastewater sequencing rather than droplet digital PCR. RNA-seq instead of PCR if you have 3 to 4 targets or more. It gets to the point where if RNA-seq were $25, the amount of time you make in PCR experiments, which are almost never really done very well because you're cutting corners, you're deciding on a housekeeping gene, or the students don't understand necessarily the complexity of CT calculation and so on. RNA-seq is extremely robust because you've got every gene you want to normalize and so on. It's just expensive, but if you get to the point where it's $25 or $30 per sample, why not? Enough with targeted panels. CT, DNA, cfDNA, that's very expensive to search for mutations, but if you can sequence deeply enough, you don't really have to worry about deciding which mutations are important. You can sequence any mutations that are there. Bulk RNA-seq, for instance, that's currently, or for 25 years, it's been the way that people have studied the transcriptome. Single cell sequencing is still very expensive, but since you can go from single cell data to bulk data by pseudo-bulking, combining it, why wouldn't you always start with single cell data if you could? I'll pause there and refer to that second principle I talked about before. The problem is single cell sequencing is not just expensive because of the depth of sequencing, it's expensive because of all of the other steps that go into it. And so the same way that Google and Amazon are worrying about electricity, we're worrying about that. Like, how do we make the rest of it inexpensive? Because it's still expensive because of sequence capture. So that ends up becoming our and your focus. Also, of course, informatics at the back end also become an issue. So these other techniques like split-seq methods like parse and scale, these can get to 1 million cells per sample, and there are now 100 million cell initiatives that are being carried out because sequencing is becoming inexpensive. The other thing you can start to do is to use the sequencer as something else. So our new Aviti has been upgraded to an instrument that sequences cells in situ. That's just been used just by Kaylee so far. She was the beta tester, but there's a seminar on January 24th where you can hear more about that. So you start to take the sequencers and you use them directly as kind of single cell devices. Also, if the sequencing is really cheap, you might start to do things like sequence entire organisms. So we've used the Ultima in some pilot experiments to sequence entire Drosophila, entire C. elegans, merely for the purpose of knowing whether or not they're clean. You're about to do an experiment on 8 lines of C. elegans and you can sequence them for $30 a piece. Wouldn't you, for or $50 a piece, for $400, know that your mutants are what you think they are? And they haven't drifted since you started your experiment, since the last time they were isolated, or God forbid, the wrong C. elegans worm was picked and you're working on the wrong thing. So you could just sequence it and that becomes your unit of analysis. It's a little bit like using Plasmidsaurus now. You don't just sequence part of your plasmid, you always just sequence the whole thing. Cell line authentication, similar. Genome editing characterization. This is all quality control. And lastly, somatic genomics. 30x is fine for a diploid genome that is the same in all cells, but of course it's becoming apparent that that's not true. I'm going to talk about a thing called PPM-seq. This is a commercial term from Ultima. It stands for paired plus minus sequencing, but also part per million sequencing because it's got an accuracy of 1 in a million. And so it allows you to start to kind of like sequence DNA in such a way that you can detect extremely rare events. And what's becoming clear through this initiative from the NIH, somatic mutations across human tissues, it's becoming clear, I heard some talks at ASHG in November, that there are these dominant effects that small clones of somatic mutations can have that might be extremely important in driving, for instance, tumor biology, but many other things, brain phenomena as well. So, to sequence something that's somatic, of course, 30x is nowhere near enough. You need 300x or 3000x or 30,000x. And this is what you start to do with sequencing that's extremely cheap. Circulating tumor DNA, circulating free DNA, fragmentomics. These are cases where you're sequencing a fragment merely to get its length and perhaps termini, and that's actually the data point you're after. And then 5 and 6 base sequencing. So we're working with this company Biomodal. That with their technology, you can take DNA reads and read all 6 bases. So ACTG, methyl C, and hydroxymethyl C. And you get it in the context of the genotype. Very expensive because you're doing a lot of extra sequencing with that technology. And then lastly, enough with mass specs. With all due respect to our friends in the Center for Mass Spectrometry and Proteomics, you can actually now use these panels of proximity ligation antibodies to start to profile thousands of proteins at the same time. The readout is next-gen sequencing. So these are all the things you do as sequencing gets cheap. And so let me now go into the main part of my talk, which is what makes this possible. So if we look at the history of sequencing, there's been 3 revolutions in some senses. The first revolution, that's a picture of a dideoxy nucleotide. The dideoxynucleotide allowed Sanger to come up with Sanger sequencing. And, of course, this dideoxy terminator stops polymerase at a base. It gives you a fragment of a size. If you mix these stochastically with natural bases, you end up getting a ladder that stop at bases. And that was the basis of Sanger sequencing, along with all of these other innovations. Over a period from 1980 to sometime in the 90s, it was refined to the point that a company finally mastered it, Applied Biosystems, and that kind of became the standard sequencer that really finished the genome project. That was revolution number 1. It was great, but it really only dealt with the final sequencing part. You still had all of this upfront cloning and clone collections, and it was very, very difficult. So the Human Genome Project ultimately, although the second revolution of next-gen sequencing didn't really help with the Human Genome Project, it certainly helped since, motivated by this need to parallelize not just the sequencing, but all those other steps. You had these innovations, and I'm not going to spend a lot of time going through them, but essentially all those other steps where you had to kind of do a lot of clonal manipulation of fragments upfront of Sanger sequencing to have individual reactions, that was all parallelized, and we didn't have to separate that. So the modern idea that you take bulk material, you make a bulk library, you put it on the sequencer, and it gets separated up into individual reads there because it's clonal all on the instrument, that was pretty much revolution number 2. We all know who the winner of that revolution was. And that has been a stable platform for a long time, but Illumina's patents ran out a couple of years ago. And that's what's really kind of been the driver for revolution number 3. And we're super jazzed about it. I mean, we've been spending most of our time trying to position the University of Minnesota and you to be able to take best advantage of this while maintaining access to the legacy instruments. So let me talk a little bit about that strategy. We've decided to outsource the NovaSeq X Plus and PacBio instruments, as many of you probably realize. And we did so because those two platforms have not been really democratic. Places like us, and we're a pretty big core facility, but still not big enough to really be able to offer those competitively compared to the Broad Institute and other places. And it got to the point where you realized, you know what, better to join them than to try to beat them. And so outsourcing is a really great option. And we do outsourcing for people. We just pass their costs along to you. The democratization that's occurring is that Ultima is coming out with even cheaper sequencers. And of course, for it to be democratic, you have to get access to it. And so that's why we've been focusing on getting access to that instrument. There's been democratization has also occurred on the Aviti platform. I'll go into this in detail, so I won't talk too much about that right now. But before we kind of got into the Ultima platform, we focused on the Element Aviti, which I think has, as I'll show, been really beneficial to people in terms of lowering costs. Diversification is the other kind of change here. If you look at what happened in the diversification of computation, what was originally a computer became all of these different applications. And that's occurring too in sequencing. So we've been focusing on these uses of sequencing, kind of like I outlined before, like the cytoprofiling on the element instrument, TPM-seq, 6-base sequencing. These are all ways in which computation gives new capabilities to you. So that's what we're focused on. And lastly, maturation. And let me just briefly touch on this. Ultima is a company built by people from Silicon Valley. And as I'll explain, all the technology built into it is essentially repurposed computer technology. It's very unique in the way that it takes on sequencing. They've kind of reinvented every part of it. But behind that, and we know this because we spent a lot of time talking to the executives at the company, their vision is that sequencing will become cheaper and cheaper and cheaper, and they're building their company around that. So they're very vertically integrated. They have very low cost for everything. And their plan is to continuously drop the cost. That's not something we've ever heard from companies before. It has been the case that Illumina, for instance, and other companies have dropped costs, but that hasn't been their explicit goal. Their goal has always been new capabilities and so on, and they've always thought as high a margin as they could possibly extract. The interesting thing about the folks here is that their experience is that if you make computation cheap, that people always use more of it, and that's been the truth in Silicon Valley. And so we think that this is the only company that kind of understands or shares this vision of arbitrarily inexpensive sequencing. And so that's why we're making a bet on that platform. Hopefully it's a good bet. Okay, a little bit on it. We really need to acknowledge Mike Gale, who's the incoming head of the Department of Microbiology and head of UMID, and then Peter Crawford and Kim Kirkpatrick in Rio. The instrument was funded by a grant from Rio and the medical school. And lastly, revolutions are never bloodless, although we've kind of moved away from Illumina as a platform. They're, of course, trying to innovate. This they announced a couple days after GenoFest, which is where this slide is from. Ultimately, what this ended up being was a bit of a meh from our perspective. It's a small sequencer. It doesn't yet compete, but things might change. So we've not sworn off Illumina forever. It's just right now we figure we can serve you better by outsourcing. Okay, let's talk about the Illumina, about the Aviti a little bit. That was our old fleet. These were the new options in 2023. We could have spent $2 million on them. We decided not to. Instead, we've been partnering. So we outsourced to Chicago and various companies for our NovaSeq X Plus runs. And we partner with Mayo for PacBio Revio runs. So that allowed us to kind of get rid of the costs of these instruments. And that's what our current Illumina fleet looks like. And what we've got instead now are 5 Element Avitis and 1 Ultima UG 100, which has only been operating for about 8 weeks. In practice, of course, we've got all these instruments because outsourcing works really effectively. So a little bit about why we went into the Element Aviti. The Aviti is called that because it uses avidity sequencing. And the idea of avidity is that the molecule that's used in the sequence readout is a thing called an avidite. So avidity, avidity, avidite. And let me just walk you quickly through how the sequencing works. The first step is that the molecules, which are circularized molecules, actually in the most recent version of their technology, you can literally just take a linear Illumina library and load it onto a flow cell. The flow cell will circularize it and carry out this rolling circle amplification. The rolling circle amplification, one key point to realize there is that unlike exclusion amplification on the Illumina system, where you basically have PCR occurring, there's no PCR in this. It's a linear amplification. And so that means that an error that gets introduced does not end up getting exponentially amplified. So one of the keys to the accuracy of the Aviti, and I'll cut to the chase, it's a much better sequencer than the Illumina sequencer in terms of accuracy, is because of this rolling circle amplification method. Then what happens is a step of avidity sequencing. And the avidite looks like this. The avidite is basically a single nucleotide. So this might be C, a deoxy C, tied to this fluorescent molecule. And you'll notice that there's 3 or 4 arms, all of which have the base. And all of those will bind the pocket of a polymerase bound to that avidite on different positions on the same cluster. So what that means is that that binding looks something like this in this cartoon. In order for that binding to work, the molecule must find 4 spots with a T on the template across from that adenine. So let's say that there's been an error introduced during the rolling circle amplification. That error is only going to be in one position. It's not going to bind to the avidite effectively. And so that's the second reason why you end up getting highly accurate sequencing. Now, mind you, this avidite does not get extended. This isn't an extension reaction. It's simply a detection reaction. And because avidity is used in the binding, you can use very low concentrations of these reagents. So it also makes it inexpensive. So the segregation of base calling from extension also permits optimization of both. The important point here is that these are native nucleotides. They don't get extended because there's no polymerase there at the time, but they don't have a di or a dideoxy terminator, so the binding is extremely efficient. In normal SBS sequencing on an Illumina, they've modified the nucleotide up the wazoo. It's got a fluorophore and it's reversibly terminated. So it's it's not a high affinity binding the way this is. At that point, you wash off the avidites, you carry out optical signal detection. This I'll skip over in the interest of time, but it simply shows that the avidite binds avidly, whereas monovalent nucleotides quickly wash off. This shows that even at low concentrations, you can get a stable signal with the avidite. Let me move on from that. And then the cycle is, the final step is to wash off the avidites, extend now with a reversible terminator, but without a die. And that can be optimized not for detection, but for extension so that it's complete. And then the cycle repeats. So the main take home there is that the resulting accuracy, so this shows you the predicted Q scores versus, and a histogram of predicted Q scores. You can see that a Q40 is 1 in 10,000, an error rate of 1 in 10,000. The very highest Q scores you get on an Illumina read are around 39, 40. And you can see here that you're getting a modal 40 Q44. So that's 1 error in like 30,000 nucleotides. So it's extremely high quality. And also it's very impressive in terms of length. So we carried out some, well, this is actually from Element, but it's highly accurate way out to 300 bases as well. And so the nice thing about high accuracy is that since accuracy always falls off with length because of phasing and pre-phasing and sequencing, if you're starting off with a higher quality, you can afford to have more of a loss over time. One of the main details is that the real errors in Illumina come after homopolymers, whereas this shows you the error rate, and you can see it's a pretty significant rate after homopolymers. On either the NextSeq or the NovaSeq, you can see that the Aviti does very well after homopolymers, and this is just the detail of as the homopolymer gets longer, the Illumina error rate goes higher and higher up to the point where it's like 7 to 10%. Now, these are errors not in the homopolymer, but after the homopolymer. And as a result, if you look at the median pileup from a NovaSeq sequencing run versus an Aviti sequencing run, this I think is the easiest way to appreciate the difference. All of these SNVs are errors. They would get resolved, of course, because in this pileup, that's a very simple thing to do. You can see that these are just errors. But what this shows you is that you can get away with much shallower sequencing in many ways, and you don't have anywhere near as many errors complicating your analysis. So for a clinical lab, this is actually really nice. You don't end up getting caught up wondering and chasing down low events. Also, you can immediately see that if what you're interested in are somatic events, this gives you the ability to detect rare events much more easily. And as I'll point out, this is actually not even their highest quality. This is their V1 sequencing. V2 is this Q50. It's just become available now. This is 1 error per 100,000 bases. And the way that they achieved this again was to simply, innovate. They looked at, this is the error rate under this new cloud break. The old cloud break. And this is the error rate using their new Ultra Q. They simply realized that, most of these errors, which are higher in cloud break than in Ultra Q, are oxidative DNA damage signals. And so they realized that by adding some steps to the library creation that cleaned up and prevented that kind of damage, when you combine their library creation with this sequencing, you can now get 1 error per 100,000. So if you're interested in really rare events, in the old days you had to spend, there were all sorts of tricks for doing this, duplex sequencing and UMIs and so on, over sequencing. Now you simply can get good quality right out of the box. And to foreshadow Ultima, there we've got 1 error in a million. And lastly, the Aviti, one of our Avitis, we've upgraded to a thing called the Aviti 24. Please come to the seminar on the 24th to hear more about it. But here you load single cells directly onto the flow cell. Haley's lab did this. And then using panels of probes that end up being read out as sequencing, you can, within inside the cells, measure proteins, phosphoproteins, various cell markers, including lipids, organelle markers, and then genes, panels, and custom. So it's another single cell technology. But all of this is to talk about the ways in which the Aviti is better. Honestly, one of our main drivers in purchasing it was because it's also cheaper and because of the way that it's configured faster. So it's one of these rare cases where, their goal here, this plot shows you cost as a function of the scale. So Illumina has always been somewhere along this line. You could get really cheap sequencing, but you just had to do tons of sequencing. What Element's done is that they've basically put a line that's parallel to that right underneath it. In other words, if you want to get really cheap sequencing along here, you don't have to sequence as high. So you can see this is about, a 10-fold less sequencing. In other words, they've made a 1 billion lane flow cell that costs the same per read as a 25 billion Illumina flow cell. In order to do this, you actually have to use a lot of their reagents. That cost is basically based on a discount. And so we saw this early on and said, okay, let's shoot to get 3 to 5 AVDs and fully replace all of our Illumina sequencing with it. That was achieved in 2024. The nice thing about having these 5 sequencers is we can run one flow cell and get 1 billion reads, or we can run all of them and get the 10 billion reads you might get on a 10B flow cell. Those instruments are now all installed, operating very smoothly. One of the instruments here is in CCRB, and that's the one that's converted to the VD24. And as a result, we now have kind of a trifecta for this paired end sequencing that you're used to from Illumina of using the AVD. Effectively, now we also briefly did some experiments where we compared the AVD to the NovaSeq just to make sure that there weren't going to be any differences in performance. To cut to the chase there, we found that, we took a large number of samples that have been run by RNA-seq on the NovaSeq, and we looked at the correlation between the NovaSeq and the AVD, and you can see that the lowest correlation and highest correlation are both very good. If you look across samples, those correlation coefficients were excellent. Same thing with microbiome performance. We did a number of analyses to see whether or not a switch from the Illumina to the AVD would make a difference. The bottom line was it didn't. Those are on our website, by the way. We recently had, so if you want to go read those white papers in detail, you can. I do encourage you to look at our news and events, and if you're not signed up for our newsletter, you can click on the link on our website and do so. Most recently, we announced, we shared this. You can see this is our Illumina sequencing. So this is all MySeq, NextSeq, and NovaSeq runs combined. You can see this kind of growth. There was plenty of sequencing over here on the HiSeq that just wasn't included in this graph. So, we've been tootling around at about 80 runs between those sequencers per month for a long time. This is COVID. It took us a while to get back, but this is the AVD. And you can see that this number is going to continue to drop, we expect. And as a result, we achieved the volumes to get permanent discounts from Element this fall. And so, these were our rates last June. These are the rates in July through December. These are the rates we just refreshed recently. And, you can see this is just a huge drop. So this isn't the free I was referring to, but it's getting in that direction. And so the point is you can all start to think a lot more expansively about your sequencing applications. This is another way to look at it. This is the cost per billion reads on like the NextSeqs. We see them as it not being a very, we still will maintain it for people who want to use it, but it's not a very competitive platform anyway. I mean, the sequencing's worse, and it's about 2 to 3 times more expensive than the AVD. But even the NovaSeq 25B lane, so this is their biggest sequencer in their cheapest output, is more expensive slightly than the AVD, which we have here and we can run quickly because we've got 5 of them. So, if you haven't considered the AVD, we encourage you to do so. All right, so, we made this joke internally that, if the UMGC were on Facebook, we'd call our sequencing relationship status available, because we used to just be an Illumina shop, but we've really transitioned now to having a lot of different platforms, and we will remain open to whatever works best. So let me talk a little bit about the Ultima UG 100. This arrived in October. It was certified by Ultima in November. We've now already sequenced close to 1,000 genomes on it. We got somebody who wanted, I mentioned that project to you. It's always good to do your testing with an external partner. They've basically been funding all of our experimentation, the breaks, the delays, and so on. Better to do that with an external partner than with you all. But so far I can report that it's working very nicely. We're getting 30x genomes. off of it. And what we're working on now is the pipelines for using Google Deep Variant and calling them. So in the next week or so, you'll see on our website that, internally here at the U, a human whole genome 30X, including variant calling, is going to be like $176. So if you're interested in doing very large scale human studies, have at it. There are some caveats, of course, that's 20 samples at a time. But still, it's going to enable people to start to really give up genotyping, give up all sorts of things, and just say, hey, I just want whole human genomes. A little bit about the technology of the Ultima. This was in 2022 that it was first announced. Ultima worked very, very closely with the Broad, who've had it since 2018 or something like that. So, and here's the key points. It's an open flow cell design. It's basically a silicon wafer of the same type that is used to cut CPU chips from. That's their flow cell. It's a large surface area. I mean, they're like this big. Standard 10 millimeter of the type that Silicon Valley makes tens of millions of a year. They use mostly natural nucleotides, like Element. They don't have highly complicated nucleotides that screw up the biochemistry. They do optical endpoint detection. I'll go into that in detail. You get quite long reads. They're single end though. It's not a paired end instrument, but you get single end reads of up to 300 nucleotides. Very quick runtime for that length. So not 72 hours, but less than a day. And the main thing is that it's about half the cost of the Illumina 25B or Aviti current. A little bit about the technology. That's a picture of the standard silicon wafers. Rather than being a closed flow cell, it's open. It doesn't have a top, a cover. They basically flow reagents on because it's spinning quite fast, and they have a pipetter that pipettes the reagents down into the middle, and it just spreads out by centripetal force. That's apparently how these wafers are coated in Silicon Valley, and they were like, we don't have to reinvent this. So by using, they've leveraged decades of this iterative cost reduction. I mean, these have been being made since the 60s and 70s. So they're really, really cheap now, and they didn't have to invent this. They're patterned. The wafer surface is patterned, so that's easy to do. That patterning is done. That's photolithography is how you make turn silicon wafers into chips. They don't have a complicated pattern. The mask that's used to make little spots that things will stick to is just lots of little holes. So that's cheap too. The key differentiator is this. Extension occurs in flow space. I don't know how easy that is to see, but what this shows is that they have nucleotides that are modified and nucleotides that are not modified, a mixture of them. So that allows them to use low concentrations of the fluorophore. The reason they do that is they don't want to wash out the signal. These are all molecules that are on the same cluster. That's what this cartoon shows. And you can see that if you have one base that's complementary to the, sorry, let me back up. They flow nucleotides one at a time. So they add, A plus fluorescent A. They wash it off. They add G, then fluorescent G, C, T, and then A again. It's not dideoxy terminated. And so the nucleotide is only added to the cluster if the complementary base is present. So if the next base on the sequence is a T and they flow an A will be added. If it's two T's, two A's will be added, and the signal will be twice as big. If there are three T's in a row, three A's will be added, and the peak size will be 3. And so unlike standard Illumina sequencing, where you add 4 bases at once and you have to distinguish, is it A, C, T, or G, which is the color discrimination. Here, if you get any signal at all, it means that the next base is the complement of the nucleotide you flowed on. And all you're really interested in is how big is that peak? The strength of that is that it's extremely accurate. You're not trying to say, am I seeing A or T? The problem with Illumina sequencers is they've got spectral overlap between those fluorophores. They have to discriminate. Here, there's no discrimination needed. If you've got a signal, it's the same signal used for A, C, T, or G. The Achilles heel, I'll get to that in a minute. So hopefully everybody understands that idea. You're getting a peak size that's different. It also means that the length of each sequence is different. So If you had a sequence that was entirely ACTG, ACTG, and so on, then the length of your read would be exactly the number of flows you had. And typically this instrument will have 400 flows. So 100 sets of A, then C, then T, then G. Obviously, if your sequence has got places where you've got homopolymers, the read will be longer because that one flow is going to give you 2 or 3 or 4 nucleotides of length. So the read length is not fixed on the ultimate. It depends on your sequence. The Achilles heel of this kind of sequencing, just like the 454 sequencer, if those of you are familiar with it, it was also flow-based, is that once you get out to a homopolymer size of, say, 7, 8, or 9, now you're trying to distinguish a peak size of 8 from a peak size of 9 with an error, say, of a third of a unit of that peak size. That starts to become tricky. And so these flow-based platforms have always suffered in homopolymer readout. So this was the original bioRxiv paper in 2022. We were like, well, that looks interesting, but not even commercially available. Then at the 2024 AGBT meeting last February, they showed this data that they had really solved the problem with homopolymers, and they solved it using AI. It's not so much, there's really nothing you can do to the chemistry to improve it. Maybe tweak around the edges with imaging, but they, like everything else, AI allows them to distinguish between those signals, probably in a sequence context-based way. I mean, this is all secret sauce that's built into the instrument, but the bottom line is they showed this by doing genome in a bottle comparisons and getting high accuracy. So you can see now that, this was really not acceptable. An error rate of, of this high for homopolymers would have really been a non-starter for clinical applications. Wouldn't have mattered so much for counting applications, but for clinical sequencing, it wouldn't have been okay. And now it's really quite great. And then they also announced the cost. And this is the thing, that improved accuracy was kind of component one in our decision. The second was this. And what they announced was it's $2,000 for one of these wafers. That wafer is going to give you 6 to 8 billion reads. And so if you compare it to any of the Illumina flow cells that you all might be familiar with, the 1.5, 10, or 25 billion, you can see that the cost commit per run is lower. That might not sound very interesting to you, but for us, it's huge. The cost commit is like the amount of money that you're going to burn by turning the machine on. And the problem with the NovaSeq is it's always been like $16,000, $20,000 per run, which of course means we have to pack the run with the perfect combination of flow cells, which means that things are slow for you because we either are going to blow half the flow cell or we're going to, and so that'll be really expensive. So it ends up slowing things down. The nice thing about this commit is that, the cost of, let's say that we wanted to get 10 billion reads for you, or sorry, if we only wanted 5 billion reads, we can run this. We'd only be using half of it, but it's still going to end up being much, much cheaper than any of these flow cells. And the loss to us, if it goes badly, if the library is screwed up something, is just $2,000. We can recover from that. It's hard to recover from $16,000. And so again, this gets back to what I was saying is when it's arbitrarily inexpensive, you start to be able to behave in a different way. We can start to be better partners because we're not freaked out about cost losses and stuff. Okay. It is targeting, the Ultima is targeting explicitly NGS hungry applications. So clinical whole genome, I already showed you. We can generate a whole genome at 30x for about $167. So if you wanted to go for 100x, say, for a clinical whole genome, that starts to become quite affordable and probably superior to endless exome capture with all of the complications that introduces. It also will be great for single cell and omics. You can see that the cost per million reads is only 30 cents. So if you're interested in, for instance, 10 million reads, it's $3. And for oncology and CFDNA, these are their applications that they're targeting. But there was one last thing that really put this instrument over, into the we must have category, and it's this technology called PPM-seq. It's a little bit counterintuitive, so I'm going to go slowly through this. Here's a picture of a perfect DNA sequence with no damage. And here's that fragment with damage. So let's say that there's a damage to the top strand and damage to the bottom strand, just for this cartoon. In PPM-seq, it's a technology that has to use PCR-free libraries. You have to take native DNA with adapters added and put them directly into the library, into the sequencing instrument, the emulsion PCR. So this strand that's captured on a bead for emulsion PCR has both strands, meaning the native actual variant relative to reference, as well as the damage. What happens then is that both strands are amplified by PCR in this emulsion droplet. It's a lipid oil droplet. And so you'll get a mixture in this instance. This is an example of a cluster where there were damaged molecules in there. Unfortunately, this cartoon comes from Ultima. They don't have a parallel down here. I plan to make one that shows the same strand without damage, but pretend there's one there. So what that would look like is just this molecule. This gets denatured and then you're sequencing, but the thing to remember is you're sequencing off copied strands that came from either A or B strands. In other words, both the original strand and its complement. What's going to happen then is that when you get to this base here, you're going to have a mixed read. You'll have two different sequences being read out, and just like a Sanger trait with two mixed reads, you're going to end up having both A and C peaks at the same place, or both T and G, or both T and A, and so on. All that they do is to throw it away. When you throw away those reads, you throw away all errors. And so in a read in which you didn't have damage, and that didn't result in a mixed base, you can be guaranteed, or guaranteed up to a 1 error per million, that that's a perfect read that didn't have damage and doesn't have a mutation introduced during the sequencing process. That means, and this is a little bit misleading here, because it's not like you base call this. You throw this data out, but the parallel read that didn't have damage would be clean. And so all you end up getting in your data at the end are clean, high quality reads. And you might think, oh, don't you have to throw away a lot of DNA, a lot of reads? No, the reads are 100 base pairs long for this implementation. With an error rate of even 1 in 1,000, you're only throwing away like 0.1% of, or 1% of reads. So you're not losing any data, but what comes through is entirely clean. So this is a picture. This is their standard old sequencing. This is what they're getting to now, is PPM-seq. It's 80% of reads have got an accuracy of 1 in a million. What this means is that you can actually now take circulating free DNA, sequence it very deeply because it's inexpensive, and you're being able to detect mutations that are there at 1 in a million. So for cancer biologists, I think that will be uniquely enabling. There's this, program that's run by the NIH using, there's multiple different approaches for doing somatic mutation detection. This PPM-seq was a late entry to that program, but what's nice is that it doesn't require any highly sophisticated molecular biology. It's just plug and play. So we plan to try that out in the next couple of weeks. We've so far only done normal native sequencing. And once that's in place, I think it'll be great for people in the Cancer Center to kind of think about playing with. I could talk about the collab now. I wasn't sure I was going to go into this last section. I think I should stop in case there are questions, because if I go through this next part of my talk, there won't be time for questions. It's probably more important that I answer any questions people have.
Are there any questions? Hang on, I'll bring microphones.
That's awesome, Kenny. Is the Supercomputing Institute going to be able to keep up with what you're going to be able to produce in terms of human genome sequencing data? Because that's not just storage, that's secure storage.
Yeah, well, if it's clinical, right, it has to be secure storage.
Even if it's clinical specimens for research use only, which is what my lab generates a lot of.
Right.
We have to be very careful about how we store that data.
Yeah, you're right. I don't know. John Garby sits on the university's HIPAA compliance committee, so I know that they've been talking about that. That's a good question. And right now, that instrument is actually sitting outside of the university's firewall, so note to self. Oh, hi, Mike.
Yeah, so I got two questions for you. So the first is with that last section where the part per million sequencing stuff. So you showed us, where there's a mismatch between the two different strands and those just get thrown out. I see how that works if you have a known standard that you're comparing it to. What if you're sequencing something that you don't know what the sequence is? How do you then figure out at these spots where there's a mismatch, which is right?
So that is not relative to a reference. So that's reference free. The primary signal would be compromised in that you'd find that rather than just getting a single base, you end up getting
Two.
both bases at the same position.
So there's no way to know which is right then using.
What'll happen is because it's in flow space, right? What it's going to mean is that every sequence downstream will now be out of phase. So it's like the quality of your read will go from high to crap and continue at that low value for the rest of the sequencing length. Because remember, it's flow based. So if you have a T, now you've got every single base is going to be out of phase then, right? So I think they discovered this by accident. I think they were sequencing and they were like, wow, there's a small number of reads that are really bad. And they realized, because the original chemistry of their library creation, yeah, I know it's counterintuitive. I mean, you got right to the problem. Their original library creation, they would purify single strands tied to the beads and then amplify. But they probably didn't always get the second complementary strand free. And so they probably saw that those reads had this problem. And then a light bulb went off in somebody's head. And they realized, wait a minute, that's not a bug, that's a feature. If we always make the library with both strands attached to the bead, we can do this error correction.
Okay, and the second question has to do with the cost per genome. So you were saying $160 per genome, but it looked like that was for a lot of genomes. So over the last couple of years, I've made a lot of CRISPR mice.
Yeah.
And I haven't done whole genome sequencing on them. But if it's only going to cost me $200 a mouse, I'm going to do it. Totally. So how much is it really going to, let's say I want to do 10 or 20. What would that cost?
So,
ballpark idea.
You're familiar with CharterSeq, right? Which we put in place for this problem with RNA-seq. Illumina sequencers didn't allow us to do a small number of RNA-seq, so we would do a run once a month that everybody could get on, and it was inexpensive because we bulked people's work. We can do the same thing here, right? So we need 20 samples at a time to get that low price.
So it's just
but
20.
You're not going to be just 20. You're not the only person who's going to have 8 mice. Other people might have another 4. And so I think what we'll do is we'll put in, just for people at the university.
But 20 would cost 2,000, something like that. Is that right?
Well, yeah, right.
Okay. We'll talk because I have a 20 at least.
Well, I mean, what we want to put in place is a service that allows anybody who's got, because, mice and human are about the same genome size, right? So those would pair together. It doesn't have to be 20 human genomes. It has to be 20 about 3 gigabase genomes. And so over time, we'll have probably shared runs for people who don't have a large enough sample, but the commit cost is really clear here. You might just say like, you know what, screw it, $2,000. My actual cost might be $300 per sample, but I can bear that because I just want to move forward. So that's the nice thing about it is that, we don't want to, every time you try to achieve these efficiencies for people, you end up pissing people off in terms of turnaround. And what's nice is both of our platforms now don't have that problem.
Yeah, thank you. I actually have a question about the clinical lab, and they do a lot of PCR, lots and lots of PCR. And do you have any sense of, how quickly is this going to come to like a CLIA certified assay where we can not do PCR anymore and just sequence stuff?
Yeah, I mean, we've done CLIA work, and so I'm peripherally familiar with everything that goes into it, but not an expert. It seems to me right now that it's things like FDA regulations that are probably the biggest barrier. If the FDA really goes ahead with their plans to kind of shut down and regulate LDTs, it'll be a much longer period of time because there's nothing stopping, Sophia and the molecular diagnostics lab from doing this technically, but if they have to put everything through the new LDT regulations, it'll take forever or might not happen at all. I mean, if they have to wait for the companies to do it. So I don't know. But, you know, I would like to put in place a service for people who don't need really super quick results to just do RNA-seq at, you know, like $40, $50 a sample. And at that point, you know, if you're looking at 5, 6 targets, TACMan, so on, you're better off just doing RNA-seq really.
Right, right. I guess it's more an issue of like, if you're trying to develop an assay for clinical applications, do we
Yeah.
give up on PCR entirely, or is
Yeah.
it? Yeah.
One thing I will say is this: if you look at the companies that have got the UG100, they're all molecular diagnostic companies. They're only, like, we're pretty much the only core. I mean, you should brag a little bit here. They have a certified service provider program. Ultima does. There are 3 certified service providers. 2 of them are companies, and only 1 is academic, and that's us. There are very few core facilities that have this, but Exact Diagnostics and, you know, Regeneron and all these companies that are doing like circulating free stuff, they have the machine. So they've got the resources to do that FDA process. And then they also do everything at one centralized facility. So it clearly is the market that Ultima is trying to build. What we've been trying to do is to convince Ultima that we have the same ability and ambitions. And we invited them out here and they saw our place in Forefront because they were like, you're not going to have space for this, because we don't want to just have 1 of these, you know, we'd like to have like 5 of them. Because right now, you know, it's broken 3 times already and it's out for a week, and you need to have more of them. But the idea is that ultimately all the effort and cost of kind of supporting 1, it's pretty much the same for supporting 4. So like with our Avides, that's when it really becomes useful. And then, you know, we're up there with the big girls and boys like the Broad and stuff. That's what I'd like to get there.
Thanks.
Here with Jaime?
Kenny, that's fantastic. We've been here about the same amount of time at the university, and I am amazed by your constant innovation and service. You guys have done a tremendous job, so thank you.
Thanks.
My question is, have you been thinking about the law of unintended consequences of the back end, right? So now it's so cheap to sequence that we're gonna get these piles and piles and piles of data. And how do we, I mean, are there solutions for manpower and analysis? MSI is not a solution. They don't have the manpower capabilities. There's just not, I mean, there are not enough people out there to actually do this. So are you guys thinking about the problem you're creating by solving another problem, or is that going to be entirely up to us?
Yeah, if I'm honest, the answer is no, we're not solving that problem. And yes, we are helping create it. I agree. Okay, a little bit we're working on it. So for instance, for the genomes, human genomes, there is a pipeline called Google Deep Variant, and we're going to put that in place to make a standard VCF, right? So we can do that, but for a lot of reasons, one, I don't have the expertise as the head of the UMGC, and MSI does, and that's their domain. For all these reasons, we're not going to move into kind of the more sophisticated analysis I think that you're referring to, where, you know, it's simple to do deep variant and to implement somebody's pipeline. It's very hard to do the kind of analysis you're talking about. So we'll always do what we can as far as implementing what's already available. I mean, I'll tell you, one of the places that Illumina is actually ahead of all the competition is that they bundle stuff. What they're moving towards is applications that are continuous from sample to clinical answer, say, right? It does seem to me that MSI does do a good job. It just takes, you know, of putting people in place. I think, you know, the university, I think as a whole, is going to have to continue to invest in that.
Yep. You know, getting deeper, cheaper, faster, that's great. And particularly for the purpose of finding needle in the haystack kind of application, like, you know, there's someone testing, you know, try to detect, you know, the viral sequence in the sewage waste kind of stuff. These are much better than PCR-based ones because PCR put a lot of bias, but which is supposed to be more, you know, bigger help to reduce the bias for those needle in haystack kind of situation. You know, each of them are using a different chemistry, and I suppose each of them has a kind of a higher or less amount of bias for sequencing. What do you think the best application, the technology to be used in the needle in the haystack kind of
Right.
situation?
We don't really know yet because we haven't run either UltraQ or PPM-seq yet. So I think maybe, several months from now, a year from now, we'll know better. And of course, these technologies can be combined with things like duplex sequencing and so on, which is, very powerful. That's like 1 in 10 to the 10th, so these don't get to that. But, I don't know if you've been following the SMART program, the SMAHT. There are, 7 or 8 labs that have been funded, the usual suspects, the Broad and so on. And they're doing, studies kind of developing these methods that work. And maybe this gets to Jaime's question, but if we've got PPM-seq in place, we'll try to put in place some of these pipelines that also carry out error correction for using things like UMIs and duplex sequencing.
All right, go ahead and finish. We've kind of run out of time here, so we'll let you go ahead and finish your program.
All right. Well, I mean, I have plenty more slides, and all I want to say about that is that principle number 2 that I talked about earlier is focusing on other things that, Google and Amazon are focusing on electricity generation. The analogous thing for us is that there are parts of modern deep sequencing protocols which are really exploding that are very hard for us to serve. So for instance, parse single cell sequencing is a difficult thing for us to offer because there's so much labor involved that it becomes expensive. So the collab, which we're always going to talk about, is a new model for us. I think we're about 9 months into it. We think it's going quite well, where we train you to come to our lab. We provide a fully equipped lab with all of the little bits and pieces you need, place to store your stuff. You can come and work for as many days as you want and carry out those workflows kind of under our watchful eyes. And then also in the middle of the night or on weekends when we're not around. And so the idea there is that that's our idea of how to make the electricity generation part, in other words, also affordable. Because if you already have grad students and they can be trained to do it, you don't need to rely on us, which will be cheaper, more flexible, could be faster. And it also protects us from the risk of some of these really complicated experiments where it's hard to separate the molecular biology from the cell biology, say, or from other aspects. So for instance, Kaylee's lab will probably, if she decides to use this VD24, be able to come in and operate it herself because she had to culture cells and then put them on the sequencer. That's a very difficult thing for us to arrange when we don't know when those cells will be ready. So that's what that part is about. We've got stuff about the collab on our website, so please follow up if you're interested. That was it.
All right, well, if there are no more questions, we'd like to say thank you to Dr. Beckman for a great seminar.